Hello and welcome to Bio40 Pre-Lecture 24. Today we're going to talk about bacterial genetics. We've actually talked about bacterial genetics a fair amount in the past. For instance, we talked about the transformation experiment that Griffith first observed, and that Avery, MacLeod, and McCarty further explored. In this section, we're going to discuss two other ways to transfer genes between bacteria: conjugation and transduction. In addition to their role in the experiments that showed us DNA as the genetic material, bacteria played important parts in the experiments that revealed the structure of the double helix, the structure of the gene, and the nature of the genetic code. Let's go over the common features of bacteria. They lack a nucleus and most other membrane-bound organelles. They typically have a single circular chromosome that is simply found in the cytoplasm. Additionally, most bacteria have a cell wall. Beyond that, it's hard to describe properties of bacteria in general. Though it's difficult to see, there's more diversity amongst bacteria than there is between plants and animals. In terms of length, they can range from a few hundred nanometers, or billionths of a meter, up to 500 micrometers, or millionths of a meter. That's over a 1,000-fold range in size. Bacteria also come in many different shapes, and can have many different metabolic processes. There are also several ways that bacteria can move, though some are incapable of independent movement. Many use structures called flagella, which spin and function like propellers. Bacteria also have some features that make genetics tricky. They have haploid genomes and reproduce asexually. But up until now, we've mostly relied on matings with diploid organisms. Even with phage genetics, we would co-infect hosts with multiple phage to create a diploid-like environment. But what can we do with bacteria? Let's start with taking a closer look at our model organism. Many different bacteria serve as model organisms for various sorts of studies, but the best studied bacterium by far is Escherichia coli. E. coli is best known publicly for its role in disease, but that's mostly the O157H7 strain. Most E. coli strains are benign gut bacteria. E. coli has been used in research since the late 19th century, and finally had its genome sequenced in 1997. Its single circular chromosome has just under 4,300 genes across 4.6 million base pairs. That's roughly one gene per every 1,000 base pairs. However, despite the extensive study, we still don't know what 20% of these genes do. Unlike humans, E. coli genomes are very efficient, with about 90% of their DNA encoding proteins. They have no introns, small gaps between their genes, and few repetitive sequences. The image on the right shows an E. coli chromosome that is burst out of a lice cell. Clearly, the DNA is very tightly packed in living cells. Now let's look at how we use E. coli in genetics. There are two main requirements we have for genetics: mutants and genetic exchange. When we were dealing with eukaryotes, we could identify mutants by visual differences. We could see mutant flies with white eyes instead of red, or pea plants with wrinkled seeds instead of smooth. But what can we look for in bacteria? Well, there are several different options. Just as we screened individual flies and peas, we can look at the bacteria one by one. Perhaps we could see mutants that don't have flagella, or that don't have the same shape the cells normally have. Additionally, cells can be stained with various dyes that can allow us to view differences between wild-type individuals and mutants, though it's not exactly easy to do this with microscopic organisms. And even once you find a mutant you're interested in, it's hard to pick up and move a single bacterium. It's much easier to observe groups of bacteria and how they grow in liquid media and agar plates. If a single bacterium settles on an agar plate, it will divide until it forms a colony of genetically identical cells. And we can look for differences between the wild type and mutant colonies. There are many differences we can look for. Some mutations can alter the size or shape of the bacteria colony. For example, remember the rough and smooth Streptococcus colonies we discussed in the Griffith and Avery MacLeod-McCarty experiments. Some mutations will cause the colonies to change colors, and there are even chemicals that can help us detect certain mutations this way. For instance, E. coli colonies plated with the chemical XGAL turn blue if they have a wild-type LAC-Z gene, but mutants that lack a functional LAC-Z gene are white. Other times, we look for whether or not a colony can grow under certain conditions. We can plate bacteria on an antibiotic that they are normally sensitive to, and only mutants that have gained resistance to that antibiotic will be able to grow. We can also screen for auxotrophs. Recall that auxotrophs are mutants that require nutrients to grow that are not required by the wild-type organism. The wild-type bacteria that don't require the extra nutrients are called prototrophs. For example, wild-type E. coli can synthesize the amino acid histidine, and can grow on media without histidine supplemented. But a mutant that has a mutation in a gene required for histidine synthesis cannot make its own histidine, and will only grow on media with the amino acid added to it. So we can isolate histidine auxotrophs by finding mutants that do not grow on a minimal media, but do grow on a minimal media supplemented with histidine. Finally, there are even ways to screen for mutations in essential genes, which are genes whose products are required under all growth conditions. This includes genes required for the most basic cell processes, such as replication, transcription, and translation. But how do you even get a mutant with a defect in a gene that's needed for survival? Let's save that one for class. So we certainly have ways to identify and isolate mutants, but what about the other thing we need for genetics? The ability to exchange genetic material. In eukaryotes, we accomplish this through mating, but bacteria reproduce asexually. So how can we do genetic exchange with them? This problem was first addressed by Joshua Lederberg and Edward Tatum in 1946. They took two different bacteria strains that were auxotrophic for multiple nutrients. Strain A was auxotrophic for the amino acid methionine and the vitamin biotin, while strain B was auxotrophic for the amino acids threonine and leucine and the vitamin thiamine. Neither strain would grow on minimal media, but when you mix them and wait a while, you get prototrophs. Some genetic exchange must be occurring. But this genetic exchange is very different from how mating works in eukaryotes. The bacterial exchange is unidirectional. A donor transfers DNA to a recipient, but only a small portion of the genome is transferred to the recipient. The new DNA is incorporated into the recipient's chromosome by recombination, which is not reciprocal. When a new allele is incorporated, the original is lost. Now, because the newly introduced DNA fragments are linear, it takes two crossover events to incorporate one into the circular chromosome of the recipient. This is an example based on the Lederberg and Tatum experiment. The fragment of donor chromosome is homologous to the recipient chromosome, so crossing over can occur anywhere along the fragment. But the only way to get a prototroph is if the crossovers happen at these two sites. If that occurs, the recipient cell will now have a recombinant chromosome with a wild-type copy of each gene, and only information on the circular chromosome will be maintained. Genes on the linear fragment will be lost. Crossing over could have just as easily occurred at a variety of other positions, like say here and here. But this recombinant chromosome would be auxotrophic for methionine, threonine, and leucine. This recipient cell would not be detected in our selection for prototrophs. It would not grow on minimal media. Next time, we'll see how we can use information like this to map bacterial genes. There are actually three types of bacterial gene transfer that exist. The first is transformation, which we've seen before. When a dead donor cell releases its DNA, a recipient cell can take it up. The second method is transduction. We've already talked about how phage infect bacteria, but we didn't mention one of their imperfections. When an infected cell is making new phage copies, some progeny accidentally get a piece of host DNA rather than a copy of the phage genome. When such a phage infects a new cell, that bacterium becomes the recipient of DNA from the original host. The final type of bacterial gene transfer is called conjugation. In conjugation, the donor and recipient cells physically join together and DNA is transferred across a connecting bridge. This is what Lederberg and Tatum observed, and what we're going to focus on next lecture. This feels like a good spot to stop for today. Next time, we take a closer look at conjugation and how it's used in mapping bacterial genes.